Good afternoon, children. Today we have successfully completed biocycle test. Here I have checked your papers. Most of the students they performed well. We also expect your response and your result much more compared with this present examination. We need each and every consecutive examination with some improvement. Whatever you are scoring, but compared with this present score, you have to improve your score in the future consecutive examinations. Henceforth, here I am happy with your performance, as well as I am instructing and suggesting you improve your performance in future examination also. Then today, let us discuss these solutions. Already I have posted key paper in the respective groups. Check this key, but the solutions now we have to discuss these objective solutions. Then question number 1: Down syndrome is caused by an extra copy of chromosome number 21. What percentage of offspring produced by an affected mother and a normal father would be affected by this disorder? Generally, Down syndrome. You see this question. Question number 1. What is the question number 1? This is relevant with the Down syndrome. Question number 1, Down syndrome. How this Down syndrome 21st chromosome is what is here is added with one extra chromosome. 21st chromosome is added with one extra chromosome. That is called as the Down syndrome. Question: what is the question was given? Mother was affected with the Down syndrome. This is a mother. Here, this is mother. Father is there. This is a father. How many chromosomes are there? 23 pairs. You see this here, it is a mother. Here is a father. It is. Here, mother was affected with Down syndrome. First pair. Here, second pair. Third pair. Fourth pair. Fifth pair. Sixth pair. Seventh pair. Eighth pair. Ninth pair. 10th pair. 11th pair. 12th. 13th pair. 14th pair. 15th. 16th, 17th, 18th, 19th, 28th, 21st, 22nd, 23rd will be there. What is the Down syndrome? Here, 21st pair is extra. One chromosome is there. This is a mother is Down syndrome. Father is normal. Father is normal means here first pair, second pair, third pair, fourth pair, suppose fifth one, sixth, seventh, eighth, 9, 10. 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22nd, 23rd pages. Now you see from this what will come here, half of the chromosomes. Suppose ova, mother what can produce here? Ova can produce. Father what can produce here? Sperm can produce. Okay. So how many types of sperms here? All are having what will be there? How many? Here 23 chromosomes only. Whatever the sperms will come, 23 chromosomes. Why this is choice here? From this pair, one will come. From this pair, one will come. From this pair, one will come. Like this here, how many singles will be there? There is a 23 singles will be there. 23 chromosomes. But here, how many types of ovas will come? 2 types of ovas. Why these 2 types of ova? Disjunction is there. Due to the disjunction, this is here, one is going to here, one is going to here. Due to the disjunction, one is going to here, one is going to here. So all are single will come. But whenever 21st chromosome is there, what is this? How it is split? There are three are there. At one side, two are going, another side, one is going. When ova is going, all are actually when ova is forming, but here 21st is abnormality, three chromosomes are there. How it is differentiated? One ova having a 21st location, two chromosomes, another one chromosome. So that your normal ova, normal sperm is fusing. How many chromosomes will be there? 46 chromosomes. Abnormal ova, abnormal sperm is fusing. How many chromosomes? 47. 21st is trisome. Therefore, here out of two, how many impacted? One is impacted. So, what is the percentage of children affected with the Down syndrome? If a marriage is held between this year, your mother is affected with Down syndrome and your father is normal, then at that case, how much percentage of children affected with the Down syndrome question? If here, what is the simple thing here? What is the simple one here? 47 chromosomes, autosomes will be there. Autosomal sex here, mother into father. Mother, what is the configuration? A plus 1 plus XX chromosomes. X chromosomes, XX in mother. Autosomal set, one autosomal set is added with what is this one extra chromosome. Henceforth, we have written A plus 1. Next year, what is the father? A plus XY. But what is the configuration of sperm? A plus X sperm, A plus Y sperm. And here ovas are all ovas having X only. But what about these autosomes? One is having autosomal set, another one is having A plus 1. So that in this case, A plus X ova, A plus 1 X ova, A plus 1 plus X ova. So that A plus 1 means 24 chromosomes. A means 23 singles, 22 singles here. These are 22 singles plus 1, 23 plus X, 24 chromosomes are there. Here A plus means 22 autosomes and X chromosome. This is normal ova, abnormal ova. This normal ova can fuse with whatever the sperm, what will be there, normal children will come here. But abnormal means what will be their choice here? Like this abnormality, like this abnormality. Out of 4, how many abnormalities will come? 2. So that 2 by 4 means what is the percentage? 50%. So what is the answer? First one here. First one is the 1 is C answer. The answer is what is here? 1 is C. That is the 50%. Question is here. If Down syndrome individual, Down syndrome mother married to normal man, then what would be the children affected with this here Down syndrome? What is the percentage of children affected with the Down syndrome? Then the configuration AA plus 1 plus XX into AA plus XY. In this case here, 50% ova, abnormal ova, 50% are normal ova. There is 100% sperms are normal only. Then this normal sperm with this first ova normal, 50% children are normal. Abnormal ova is there. This abnormal ova 50% means children are 50% impacted. This is the answer. See, first one, answer C is correct. Question number 2. Drosophila, sex determination. Here, question number 2 is relevant with the Drosophila sex determination. In the Drosophila, how sex is determined? Bridges gene balance theory. Bridges gene balance theory is there. In Drosophila, sex is determined by the Bridges gene balance theory. What is the Bridges gene balance theory? Number of X chromosomes, the ratio between the number of X chromosomes by what is this here? Number of autosomal sets. What is the Bridges? Number of X chromosomes, the ratio between the number of X chromosomes. The ratio between the number of X chromosomes by number of autosomal sets. Number of autosomal sets. That which will be called here Bridges gene balance theory. The ratio between number of X chromosomes by number of autosomal sets. This ratio is suppose 1, that is called female. This ratio is the 1. 1 means what is that here? Suppose here 2 X chromosomes are there, 2 autosomal sets are there. Then what is this here? It will become 1. So the ratio between number of X chromosomes by number of autosomal sets. Then if it will be 1 means what is this here? That individual is the female. Suppose it is 0.5 means what would be the 0.5? That is the male. More than 1 is there that ratio. The number of X chromosomes and number of autosomal sets. If it is a more than 1, how it will be called here? Super female. This is called as the super female. Suppose here this is a less than 1. How this is called here? This is a super male. Sorry. Here more than 1, 0.5 and more than 1. And in between, what is this? 0.3 to 0.5. In between 0.3 to 0.5, super male. This is the super male. That means here less than 0.5. In between 0.3 to, or simply you can write here less than 0.5. This ratio is the 0.5. In between 0.3 to 0.5, it will be or less than 0.5 super male. More than 1 super female. 0.5 here male is there. 1 is there, female it will be there. So here 0.62 in between what would be there? 0.6 to 1. In between here what is this here? 0.6 to 1 up to 1. In between less than 1. That means here more than 0.6 less than 1. It will be called here intersex. That means not male, not female. In between 0.6 to 1, that can be called as the intersex. 0.3 to 5, it will be called here super male. Like this here, number of X chromosomes by number of autosomal sets. They are giving values. As per these values, we are deciding the sex of Drosophila. So here, what is the question? The question Drosophila sex is determined by the, what is this answer? The ratio of number of X chromosomes to the number of autosomal sets. Which option it was given? This option is the, what is this here? A option. Option A is the correct. This is second one. Here 20 will be there. First one, what is the first option? C. And second one, what is the second one? A. C is there. Here. 1C, 2A. And we will get this here. What is this? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, here 16, 17, 18, 19, 20. First one C, second one A. And now we go for third one. Which of the following is an example of pleiotrophy? Phenylketonuria, sickle cell anemia. Pleiotrophy means one gene control. Third question. This is relevant with the pleiotrophy. What do you mean by pleiotrophy? One gene control many traits. It is a phenomenon in which here one gene. This is controlling many traits. One gene control many traits. How it will be called here? Pleiotrophy. But here, according to following options here, what is the option? Option A, hemophilia. Option B, thalassemia. Option C, sickle cell anemia. Option D, color blindness was given. But what is this here? Pleiotrophy, sickle cell anemia. Why sickle cell anemia? It is a gene, sickle cell anemia gene. What is this here? Sickle cell anemia gene here? Hemoglobin. Here, hemoglobin S is there. Normal is hemoglobin A. Here, abnormal is hemoglobin S. Like this is there. Here, this is the gene. If A is there, normal hemoglobin, it will be there here, rounded shape. But sickle cell hemoglobin, it will be there like this. But this shape has been changed. Only shape only changing, a quantity of hemoglobin is also changed. One character means only shape of RBC, one character, gene. But here, suppose you see capital T. What it is deciding? Height. But here, what it is deciding? Shape is deciding, quantity of hemoglobin deciding, and efficiency of oxygen transport decided. So one gene, what it is controlling? Many traits. Here, if only shape is deciding, then the answer is the not pleiotrophy. But shape is decided. Shape is one character controlled by hemoglobin A gene. But A is modified into S. It turned to the sickle shape. Shape is changed. Next one, second one, what has been changed? Hemoglobin quantity changed. Third one, what happens here? Oxygen transportation is changed. So which of the following character? It has been example for the pleiotrophy. Here two examples may be given. What is this here? One may be here sickle cell anemia. Another one, phenylketonuria, is also example for pleiotrophy. But in the following actions, they are not given PKU. Therefore, what is the answer here? Sickle cell anemia is the answer. Which option it is? Third one, C option. So answer what is this here? Third one, C answer. And fourth one, what is this here? There are genes. ABC genes are there. Fourth one, this is relevant with the chromosomal map. This question, where it is relevant here? Chromosomal map. In this chromosomal map, how many genes are there? In this chromosomal map, 3 genes are there. A gene, B gene, C gene. You see this here, I am giving data. Here 3 genes will be there. A gene, B gene, C gene. But what is the distance between the gene? You see in between A and B. Question number 4, what they have given data here? ABC percentage between A and B 20%. The crossing over percentage 20%. The percentage of crossing over. What is the crossing over percentage? Here 20% in A and B. Between A and B crossing over 20%. Next one, B and C 28%. What is the crossing over percentage? B and C 28. Next one, third one. What is the data given in the question? And A and C 8%. A and C 8%. This is the data is given. So there are 3 genes are there, ABC gene. Percentage, crossing over percentage given in the question only. That is A and B 20%. And here B and C 28%, A and C 8%. Find out the sequence of genes and chromosome. You have to what I told here, first far distance genes you take. In the given data, what is far distantly arranged here? B and C. Far distance, what is here? 28%. B to C, how much crossing over? 28%. The percentage of crossing over, it is directly proportional to distance between the genes. What is the distance between the genes here? B and C, 28 Morgan units. Or crossing over is 28%. Next one, what is the next distance? What is this here? 20 B and A, 20 Morgan units are 20%. And B and A means somewhat, what is this here? Year to year, next distance. What is the least distance? A and C. What is this here? 8. Year to year, 20. Year to year, 8. Total year to year, 28. You see, first one, B and C, 28. We have taken. Second one, B and A, 20. But A and C, 8. So B to C, what is this here? 28%. So what is the arrangement here? BAC arrangement. They are given what is the zigzag, whatever the question actions, whatever it may be given. Some genes are there on chromosome. They are given percentage of crossing over. According to percentage of crossing over, we can what we have first long distance, next long distance, finally least distance. First long distance where it will be there. Here first long distance here BC genes. BC genes how much distance? 28%. Next second AB. So first BC 28. BA 20 and ANC 8. So year to year 20, year to year 8, 28 total came or not. So that year, what is the sequence? BAC answer. They began, what is the answers are given? You see here BAC, ABC, ACD, none of these. BAC answer. BAC, which option it is their answer? The answer is the sequence of BC, BAC. So answer, what is the answer? BAC, fourth one is the A answer. Fourth one, A, it is relevant with the chromosomal map. And next one, on selfie. Fifth one. This is a fifth question. What is the fifth question? You see, unselfing of a plant of F1 generation with genotype. This is a F1 generation plant. They have given fifth question, F1 generation plant. What is the F1 generation plant genotype? Capital A, capital B, small b, capital C, capital C. This is a plant. This plant is self A, B, C, like this. On selfing of F1, F2 produced. Now here I am Punnett square chart I am taking. From this year, how many types of gametes will come? Two types of gametes. What are those two types of gametes? Capital A, B, C. Capital A, small b, capital C. Next year, what is this gametes? A, B, C. Next, what is this here? A, B, C. So you have to make selfing capital A, capital A, capital B, capital B, capital C. Next year, capital A, capital B, small b, capital C, small c. Capital C, capital C. Next one, you make a cross capital A, capital B, small b, capital C, capital C. Then you have capital A, small b, capital C. Phenotypically, this is. These three are phenotypically same. Why? Dominate. It is also dominate. This is a dominate character. Here, dominate character. But here is also two genes dominate, one gene. Here, recessive here. Completely, this character is recessive. So, phenotypically, it is somewhat different. So, what is the phenotypic ratio? These three are same. This is one is different. Phenotypic ratio, what is this here? 3:1. But genotypic ratio, it is a question. If the question is phenotypic ratio is given 3:1. But if the same question, it would be asked for genotypic ratio. What is the genotypic ratio? 1:2:1. What is the question? The given question is this genotypic plant is selfed. Selfed means here within the same genotype cross is made. From these two type of gamete. Total 4 individuals will come. But out of 4, phenotypically, what ratio they are showing? 3:1. Genotypically, what ratio showing? 1:2:1. So, what is the 3:1 ratio? Option A. Option A 3:1, B 1:1, C 9:3:1, and D 27:9:9:9:3:3:1. So, what is the answer? Fifth one, A answer 3:1. This is the answer. Now you go for sixth one. What is meant by dominant gene? Sixth one. What is meant by dominant gene? Suppose here capital A, the tall. What is this? Dominant character tall. What is this? Dominant character tall. But here you see genotype. This is both are dominant alleles. This is one dominant, one recessive. So what is the dominant character? The gene which expresses dominant character at homozygous condition or heterozygous condition. Both condition it is expressing the dominant character only. That is called as the dominant trait. What is meant by dominant trait? A trait which is expressed either what would be there at homozygous condition or heterozygous condition. Both cases it expresses the dominant character. That can be called as a dominant gene. So in which option it is given correct one? It expresses in only homozygous state, not. It expresses in heterozygous state, not. It expresses both in homozygous, heterozygous. At both cases, homozygous condition, heterozygous condition, it expresses its character, that is called dominant. So where it is given, sixth one is the C option is correct. Next one, seventh one. A diseased man marries a normal woman. Man married a woman. Something here, one man is there. Is a diseased man. Here, woman is the normal. Then they get 3 daughters and 5 sons. They get how many daughters here? 3 daughters. And 5 sons they got. 3 daughters and 5 sons. These daughters are deceased. Sons are normal. But what is the case here? Sons are the normal. Man is a deceased. Man is deceased means what is this here? Man is suppose X, Y. Here it is. Sons are normal. Sons always what their chromosome they are getting? Y chromosome. Okay. So now you see this is here from man it is a diseased normal woman. From woman what chromosomes they are getting? Here, like this, Babu, man configuration is the XY configuration. Disease is there, X linked. Suppose here, I am telling. But it is X linked dominator recessive. How we have to see man into woman? Woman. What is the woman? XX. XX woman condition normal or problematic? Normal woman. A marriage is held between deceased man, normal woman. But here they gave birth to 3 daughters and 5 sons. Sons are normal, daughters are deceased. But in this, why? What is the cause? It is what kind of disease? Can you tell? So you see this here, these woman is there. First here, daughters. Daughters, how they are getting disease here? Daughters means here, X chromosome, 2 X's are coming. Normal X chromosome come from mother, and here abnormal X chromosome from father. But this daughter is impacted or not? These daughters are deceased. When they will be deceased? One gene is expressing its character, that is the dominant. Normally, if here 2 cases affected gene is there, they are expressing disease means recessive character. But 2 cases here, only 1 case affective gene is there, but expressing disease means what is this character? Dominant character. Now you see at homozygous or heterozygous condition expressing his character. What is this case here? This is here dominant character. So here what is this? It is a X-linked disease. But X-linked recessive or dominant? Dominant. Why dominant? Only the gene, the defective gene present on X chromosome, but they got a disease because of it is a dominant disease. Simple thing. Here you see from mother they got normal X chromosome, father they got defective X chromosome. Even the defective gene situated on father X only, they are expressing character. So that the given disease, what is this here? X-linked dominant disease. What is the 7th one they given? Here, if a marriage is held between affected man and normal woman, they got 8 children, 5 sons are normal, 3 daughters are diseased. But what kind of inheritance pattern it is? What is this? Sex-linked dominant, action A, sex-linked recessive, action B, sex-linked character, autosomal dominant. What is this here? Sex-linked dominant. So answer 7th one, what is this answer? 7th one is A. Next one, A. Eighth one. Which of the following is correct match? Down syndrome, 21st chromosome, sickle cell anemia, X chromosome, and hemophilia, Y chromosome. Parkinson, X and Y. You see Down syndrome, 21st chromosome, correct. Sickle cell anemia, autosomal. Hemophilia, X-linked, not autosomal. They are given hemophilia, Y chromosome is given. Hemophilia, X chromosome. Parkinson, autosomal, not X and Y. So therefore, what is the correct option? Down syndrome, 21st chromosome is the correct. A is the A answer. Down syndrome, 21st chromosome, A is A. 9th one, 2 non-allelic genes produce the new phenotype when parents together but fail to day independently. Then it is called 9:7. You see the complementary gene. Actually, 9th one. What is the 9th one here? 9:3:3:1. Here, non-allelic genes. What is the complementary genes here? The combining of non-allelic genes produce the same effect. Those are called as the complementary genes. Complementary genes, what is the proportion? 9:7. Actually, this is one type of allele. This is one type of allele. This is one type of allele. These are the non-allelic genes. Non-allelic genes, both are all together producing same effect. That gene inheritance of it will be called complementary genes. But what they are given? Epistasis, polygenes, non-complementary, complementary. So what is the complementary genes are? 9 is D. And next one, you see genotype closely. A cross is happening. AB. And here, what is this here? AB. And between crosses happens. But in this cross, this is one parent, any one parent, in between crosses happen. What is this question? 10th question. A cross is happened between this genotype. But the condition AB are linked genes. But AB linked means here it is one chromosome, it is one chromosome. Here AB genes are there, AB genes are there. But here, this is one chromosome, this is one chromosome. Here, what will be there? AB genes are there, AB genes will be there. This is A, B only. So, homozygous condition, you take cross, you can take linkage, whatever it may be, what type of gamete will come? AB only. But here, how many types of gametes will come? AB or linkage. What is the condition? AB linkage. AB linkage means what will come gametes? AB gamete only coming. AB is linkage. What gamete will come? AB gamete will come. There are two kinds of gametes. Here these AB genes are linked means without exchange as these comes to gamete. Without exchange as these comes to gamete. Here sperms are only one type of sperm or any one gamete. Maybe it is mother plant or father plant. There is no criteria in reciprocal class. Here only how many types of gametes will come? One type of gamete. Now this gamete is fusing. What will come? A, B will come. This gamete is fusing. What will come? A, B will come. So what is this offspring's result here? Here A, B. Capital A, small a, capital B, small b. All are small a, small b, small b. So what is this here? This answer where it will be there? Option 1, option 2, option 3, option 4. Option B. 2 option is correct. You see, this is one. Capital A, capital B, small b. Small a, small b, small b. Which option it is there? Option B it was there. So answer what is this here? 10th one. B is the answer. Correct. Option C. When dominant recessive alleles express altogether is called recessive alleles. Question number 11. What is the 11? This question number 11 relevant with the codominance. Why codominance here? Both alleles are expressed equally. The two different alleles of a gene expressed equally. How it will be called here? This is called codominance. You see the question, when dominant and recessive express itself together is called. Both dominant and recessive together expressing. The expression of two alleles of a gene equally, how it will be called here? Codominance, human blood group. Options what they are given? Codominance, dominance, antidominance, pseudodominance. So answer what is this here? Codominance. Therefore 11th what is this here? A answer. And 12 complementary genes. Previous question already we referred. The 12th question, what is the complementary gene ratio? 9:7. Complementary gene ratio 9:7. Non-erotic effect. It was already discussed. So answer 12th what is this here? D answer. Next year, independent assortment genes does not take place. Whenever independent assortment does not take place, you see question number 13. What is the question number 13? This is relevant with independent assortment. Independent assortment. At what condition independent assortment does not take place? You see this independent assortment, genes are located on homologous chromosome. Genes are linked and located on same chromosome. Genes are linked on non-homologous chromosome, all of these. Here, whenever independent assortment does not take place here, located on homologous are linked and located on same chromosome. Here the criteria is linked. Whenever this is a chromosome, this is a chromosome. Here A gene is there, B gene is there, A gene is there, B gene is there. Maybe these A and B genes present here, but without a linkage, there is a choice to get AB crossing over. There is a choice to get AB. But what is the criteria? Those should be linked. If they are linked, no independent assortment. The question, at what condition the genes are not showing independent? The genes should be present on same chromosome and those should be linked. At that condition, they cannot show independent assortment. What is the condition? Here, to escape from independent assortment here, they should be present on same chromosome, then they should be linked. Then independent assortment does not occur. So here, what is the answer here? The question number 13, genes linked and located on same chromosome. 13 is the B here. Whenever occur here, independent assortment. Whenever does not occur, whenever they are linked. When the genes are linked present on same chromosome, independent assortment does not occur. So what answer 14th one here? This is the 13th one is the B. This is the 13th one, B answer. Next one, 15th one. What is the 15th one? Bar bodies. Sorry, 14th one. 14th, 13th one B. Next one, 14th one. 13th one B, independent assortment does not occur when the genes situated on same chromosome and linked. Independent assortment does not occur. 13th B. Next one, 14th one. What is the 14th one? You see, sickle cell anemia. What is the sickle cell anemia? Change in the amino acid sequence in beta globin chain. The amino acid sequence, what is this? This is relevant with the sickle cell anemia. Sickle cell anemia. In beta globin chain, valine, histidine, leucine, threonine, proline, glutamic acid will come. First position, second position, third position, fourth, fifth, sixth. In place of normal hemoglobin, beta hemoglobin, then beta chain of hemoglobin, glutamic acid is replaced by valine. So here change of amino acid. Okay, then what would be there here? This rounded shape hemoglobins will turn to this sickle shape. So which of the following statement is correct, relevant with this here? Sickle cell anemia. Options you see. Change in amino acid in A chain. A chain means alpha chain of globin, not alpha chain. Change in amino acid in beta chain. Yes, it is a beta chain or B chain. So in the beta chain or B chain of hemoglobin, then in place of glutamic acid, valine is replacing. Then rounded shape hemoglobins are turned to sickle shape. That is the sickle cell anemia. So what is the appropriate answer? B is the answer. 14th one, what is this here? B answer. Change of here amino acid sequencing in beta chain or B chain. What is the 15th one? The question 15 relevant to the bar body. What is the bar body? Inactivated X chromosome. Here in the body cells of man, body cells of human beings here, the body cells of human beings, not sexual cells here, body cells of human beings, the inactivated X chromosome is called as the inactivated X chromosome. How this inactivated X chromosome, it can be called as the bar body. Bar is a scientist name. Murrah Bar discovered this. Here inactivated X chromosome. But inactivated X chromosome means out of 2 X chromosomes, one is inactivated. So for dosage compensation, inactivated X chromosome is called as the bar body. For dosage compensation, one X chromosome is inactivated. So here n-1 principal we are using. N means number of X chromosomes. Number of X chromosomes. Suppose here, what would be the number of X chromosomes here? 2 X chromosomes are there. -1. How many bar bodies here? 1 bar body occurs. 3 X chromosomes are there. 2 bar bodies are there. So simple number of bar body means what? How can we find out number of bar body? Whatever X chromosomes are there, from total number of available X chromosome, we can make -1. 5 X chromosome means 5 - 1, 4 bar bodies. 10 X chromosome means 10 - 1, 9 bar bodies. What is bar body? Inactivated X chromosome. Generally, how many bar bodies are there in male individual? No bar body. Why? 1 X chromosome is there. 1 - 1, 0. In human female, how many will be there? Normal human female. 2 - 1, 1 bar body. Like this here. Suppose what is the question? They have given 15 number of bar body here in this condition. People, quadruple X. 4 X's are there in female. How many bar bodies are there? Number of bar bodies in the female having 4 X chromosome. 4 - 1, 3 bar bodies. Whatever X chromosome is there, from available X chromosome -1, they will find out the bar body. 4 - 1, 3 bar bodies are there. So what is the 15th one here? This is C is the correct one, bar bodies. And 16th one. What is the 16th one here? You see, male XX, female XY, sometimes occur. Actually, male is normal condition, male XY. Normal condition, female XX. But different question given. Sometimes, what is the case? Male has XX. Not because sometimes happen due to mutation. Female what has here? XY. Male has XX, female has XY. Why? What is the reason? Can you tell here? Transfer of segments in X and Y. The transfer of chromatid material between X and Y. Then what happens here? Suppose here, then X become the Y become the X. If X chromosome genes are going to Y will become X. Y genes are going to X means X will become Y. Therefore, instead of XX, what become here? XX. Why? Because sharing of chromosomes here. How it is actually this is a normal X. It is abnormal X. Why it is shared the chromosome? What is here from X chromosome individuals? Here XY is there. This is genes are going to here. Then this Y will become X. These genes are going here. X will become Y. Therefore, instead of here XY, male will become XX, female will become XY. So, due to the exchange of segments of chromatids in between X and Y chromosome, it happens here. So, here transfer of segments in X and Y chromosome. It is a deletion, a transfer of segment, hormonal imbalance, aneuploidia, transfer of segments. Due to the transfer of segments in between X and Y chromosome, what would be there? Male will become XX, female will become XY. So what is the answer? 16th one. This is a B answer. And 17th, probability of a 4 sons in a couple. Here, this is a parent. Father is there, and mother. Father and mother. Here, there is a sons will come. There is a offspring will come. But this offspring at first generation, there is a daughter or son. What is the probability at each generation to have a daughter or son? 50% choice. Every case, every generation, maybe like a, if you toss a coin, what is the probability to get it at 50%? Tail 50%. Like this, every generation, what is the probability to have a son? 50%. 50% means son is a 50%, daughter is a 50%. But second generation, 50%. Every third generation, 50%. Fourth generation, 50%. First time may be son, second time may be son, third time may be son. A fourth time may be son, 10 times may be son, may be possible. So if up to 4 generation, what would be the possibility? First time 1 by 2, second time 1 by 2, third time possibility 1 by 2, fourth time 1 by 2. So here 4 times son means 1 by 2 is 4 times you can write. 10 times means 1 by 2 into 10 times. So what is the probability? 1 by 2 into 1 by 2 into 1 by 2. What is the question here? Here probability of 4 sons to a couple. 4 sons means first generation 1 by 2 probability, second 1 by 2, third 1 by 2, and fourth 1 by 2. So here 4 times means 1 by 2 into 4 times. What is the answer? 1 by 16. Numerators you can all are 1 only. 1 1 just 1, 1 1 just 1, 1 1 just 1. 2 2 just 4, 4 to the 8, 8 to the 16. What is the probability to have a son for 4 times here or 4 sons here? 1 by 16. So answer is, what is the answer? 1 by 16. Where 1 by 4 option A, 1 by 8 option B, 1 by 16 option C, and 1 by 32 option D. So what is the answer here? This answer is 1 by 16. 17th is the C answer. The 18th one. What is the 18th one? This is a cross. And what percentage of tall and red flowers? Here, 18th question. Question number 18, you see. This is the question number 18. Cross between capital T, capital R. This is tall red. Tall plant, red flower. Into next one, it is dwarf white. This is the dwarf white flowers. What percentage of tall red? Here, dwarf white. This is what is the configuration? Dwarf white. A cross is made between tall red and dwarf white. Then what is this here? What is the percentage of tall and red flower? In this cross here, what percentage of tall red happens? Here, if a cross is made between tall red and dwarf white, what percentage of plants they have a tall red? So how many types of gametes will come from this? If you have to Punnett chart here, this is a Punnett square chart. From this parent, how many types of gametes? Capital D, capital R, capital D, small r. 2 types of gametes. From this, how many types here? Only 1 type. Always here you take here, whatever this here, only 1 type will come. From this here, how many types will come here? Small d, small r. Only 1 type will come from this here. So you can make a cross from here, capital D, small d, and here capital R, small r. Next one, what is this here? Capital D, small r, small r. Next time you take same will repeat here. So no need. You count for 2 or 4 or no need here. but result is same. Here like this same, like this same. Suppose out of two, how many reds are there? This is a tall red. This is called as tall red. But this is called as a tall white. We need one client here, tall reds here. Out of two clients, one is the tall red. You count for four. This is also tall red. This is also tall white. So that out of four, how many tall reds? Two. 2 by 4 or 1 by 2, answer is same. Because it is the same gamete, you go for 4 clients or 2 clients are enough here. That is our wish. So that here, out of 2 clients, how many tall reds? 1. Out of 4, 2. So what is the percentage? 1 by 2 means 50%. A cross is made between tall red and dwarf white. What percentage of plants are showing the tall red here? This is gametes from this plant. This is gamete from this plant. We make a cross, then tall red, so many will come here? 1 by 2 probability. If you convert this percentage, what is this here? 100 by 2. 100 by 2 means what is the percentage? 50%. So what is the options are given? 18 to 1. Option A 50%. Option B 75. C 25. D 100%. So A is the 50% is the answer. A answer 18. And 19th one. What is this here? According to Mendelism character, which character show dominance? Already here 7 pairs of contrast character we discussed. But you see terminal flower. Terminal flower not dominant, recessive. Option B green color in seed core. Here what is this here? Yellow color is the dominant, not green color in seed core. Wrinkled seeds, recessive. Green pod color green is the dominant. According to given option, what is the answer? 19th one. D is the correct answer. And 20th one, Mongolian idiocy. That means here the addition of extra chromosome to 21st chromosome is called as which syndrome? Already first question only we discussed here. What is here? Addition of chromosome, extra chromosome to 21st chromosome is called as which syndrome? Down syndrome, Turner, Klinefelter, triplets. What is this syndrome here? This syndrome is called as the Down syndrome. 20 is A. So these are the 20 questions today. So this is here your cycle test 1. Next cycle is molecular basis of inheritance here. So thank you one and all.